Hello again, grade 12s. This is the video lesson for the last bit of the course, which is 10.3 Gene Pools and Speciation. I'll try and keep this one a bit shorter so I can get it onto YouTube. Okay, to find the slides for this, you will go on the website as normal, and Unit 5, which is Nucleic Acids and Evolution. If you go right down to the bottom there, it's 10.3 Gene Pools and Speciation. Okay, there is a really good video from Bozeman Science. He's not an IB teacher, but he's an AP teacher, an American curriculum teacher. It's really good at explaining speciation, so I do recommend that you watch that video. And we've got the normal slideshow over here. As with the other recording, if you could just pause every now and again, write down your notes. You don't need to draw and annotate diagrams as much in this one, but there are some graphs that you'll have to draw, so it might be a good idea to make sure that they're sketched in your notes as well. Okay, so we're looking at gene pools and speciation. Essential idea with this is that gene pools change over time and therefore you get new species. An example here, we've got two butterfly species, Heliconius genus above, and they've only recently diverged. And consequently differ in 12 regions of their genome. Okay, so those 12 regions are going to mean that they might have a different chromosome number and therefore they can't interbreed anymore. Okay, other species of the genus diverged earlier and they've got more genomic changes. So these are the understandings, applications and skills, bit of a smaller one this time. We're going to be talking about what a gene pool is. Evolution requires that allele frequencies are going to change over time, looking at different reasons for reproductive isolation. So it can be temporal, that's to do with time, behavioral or geographic. Then we look at different types of divergence of isolated populations. It can be gradual, but there are other theories that suggest it can be abrupt or quick, and that's called punctuated equilibrium. We'll also look at 3 different examples of directional, stabilizing and disruptive selection, and then we get to talk about onions. All right, so speciation in the genus Allium by Polyploidy, so the onion family, which is onions and garlic and things like that. Okay, polyploidy will be a word that comes up as well, and then we're going to look at some allele frequencies of geographically isolated populations. Okay, you need to know these definitions. All right, so a gene pool is a total collection of different alleles in an interbreeding population. Allele frequency is the proportion of all copies of a gene that's made of a particular gene variant. Okay, so remember that genes can be classified as alleles if there's a slight difference in the DNA. Okay, for example, if a recessive allele made up 2% of the total number of alleles in a population, then the dominant allele, the capital letter, would make 98%. Okay, so think of that in terms of blue eyes and brown eyes. I don't know what the figures could be, but if 75% of people had a dominant allele, then 25% of people would have the recessive allele. Okay. The frequency isn't actually expressed as a percentage, it's expressed as a fraction of 1. Okay, so if 2% would be expressed as 0.02 and 98% would be expressed as 0.98. If you add both of those together, all right, the frequency will always be 1. All right, as long as it's a characteristic determined by 2 alleles, but that's all you'll be asked about in terms of these definitions. What happens if you have new combinations of alleles? Then you can have a new phenotype, okay? And a new phenotype caused by mutations can be selected for or against by the environment, depending on whether it's a genetic advantage or not. That can then lead to an evolutionary change in the species. Okay, so these are graphs that you might want to just make a copy of. All right, if we have an original population, if there's no selective pressure, if there's no change in the environment, then the population will not evolve. You will have some members of the species that have a certain phenotype, a few that have a different phenotype, and the vast majority will have the phenotype in the middle. Okay, we're used to saying things like that. Imagine it being 2 heterozygous parents for fur colour. All right, you would find that most of them are heterozygous, so they would have the kind of middling fur colour, and a few would be homozygous recessive and a few might be homozygous dominant. Okay. Directional selection means that there is a selection pressure that does something to make being this lighter coloured fur, for example, a disadvantage. Therefore, the population is going to shift towards the other end over here. There's stabilizing selection, where there are two selection pressures. Okay, so one for the extreme on one side and one extreme for the other. So the population moves to a more kind of middling value. Okay, directional selection, where the original population gets split, a selection pressure splits them in the middle, and then you get one population of one type of allele and one population with another. So their phenotypes are going to be quite different then. If the selective pressures do change, then the population will evolve, but how it evolves depends on which phenotypes experience the greatest pressure. Okay, so which characteristics have become a disadvantage, and those particular members of the species will die out. We're going to now go through, so there'll be an example of each one of these. So, directional selection. Okay, an example of this would be the medium ground finch. All right, one of the finches that was found by Darwin. Okay, so during dry years, small seeds are not abundant. So instead of fur colour being here, this would be beak size and beak shape. Okay, going from smaller beak shape over here, better at picking up the smaller seeds, to larger beak shape over there. Okay. So during the dry years, small seeds are not abundant. Therefore, the population is going to shift to the right, and these will be the species that have the bigger, more stable beads. So the birds with the larger or the tougher beaks become more frequent. Okay. Stabilizing selection. Okay, so in this situation, it's better to have the medium, the middle characteristic. Okay, human birth weight is a good example of this one. So the selective pressure would be babies that have low birth weight over on this side. They lose heat more quickly and they get ill from infectious diseases more quickly. So it's not good for babies to be too small. So that's why it's dangerous for babies to be premature. Babies of large body weight are more difficult to deliver through the pelvis, which causes problem for the babies and therefore problems for the mother as well. The result is that medium weight babies have a much lower mortality, and hence the frequency of medium weight babies is going to increase. Disruptive selection, probably less common, this one. So, for example, the selective pressure in this situation might be that the soil close to a mine working, so a mine where you dig for metal or coal or whatever else, might be contaminated with metals, for example copper. The result would be two distinct grass populations, so a slower growing metal tolerant population, okay, over here, and then a faster growing population that are not as tolerant to the metal. Okay, but the medium growing ones wouldn't grow anymore. Just referring again to the video, it's on the website. Okay, if you look at the presentation, it's embedded into it as well. So if I press that, that would play, but I'm trying to keep the video short, so I won't do that. Okay. Next understanding we're looking at is reproductive isolation of populations can be temporal, to do with time, behavioural, or geographic. Okay, this is only in sexual reproduction, so it's only when there are two parents. Okay, it doesn't happen in single-celled organisms, so sorry, bacteria. Temporal, to do with time. Okay, so Pinus radiata and Pinus attenuata are prevented from hybridizing. So that means making a kind of new species of the two different seeds because they have separate pollination times. They can be made to hybridize by pollinating them manually. Okay, so if farmers intervene or plant growers intervene, then you could make a new species or a hybrid species there, but left on their own, because they pollinate at different times, they will not interbreed. Okay? Ecological isolation. So in this situation, it might be that this particular viola really wants to say that they prefer a calcium-rich soil, and this one over there, the tricolor, might prefer a magnesium-rich soil. Okay, so that just means that they would not be flowering at the same time. Behavioural isolation, this one is quite interesting. So animals exhibit courting behaviour. So they do songs and they do dances, or they might release pheromones to attract mates. Individuals are only attracted to and will only mate with members of the opposite sex who perform the appropriate ritual or release the correct chemical. So these species doing these dances, they can't just go off-piste and do their own thing. They've got to be able to do the dance that works for their species. Okay, so these behaviours that they exhibit, they just don't work, or they might sing a different song one day, okay, and that song will not work. Okay, last little bit here. You might be asked to compare allele frequencies for geographically isolated populations. All right, so around here, that's the British Isles over there. You've got Scandinavia, Iceland and Greenland, maybe. Okay, I think so. And we're looking at 2 alleles of the gene, code for versions of phantophycin, which must just be a particular protein. Oh, here we go. It's an integral membrane protein called phantophycin. Okay, these are in codfish, and they were collected in 23 populations and tested to find the proportions of the alleles in each population. Remembering the frequency can go from 0.0 to 1.0. The total frequency would always add up to 1. Okay, so you can see there will be some that only have one particular allele. All right, and over here where there's a little pie chart situation, you can see that there's 2 alleles. Okay. So, question. State the 2 populations with the highest pan-IB allele frequencies. State the population in which the allele frequencies were closest to 0.5. And deduce the allele frequencies of a population in which half of the codfish had genotype pan-IA, pan-IA, and half had the genotype pan-IA, pan-IB. So here you might want to do a little Punnett square and figure out what those population frequencies would be. So just to go back, so pan-IA is the grey part, okay, and pan-IB is the darker part, okay. So if you could just pause the video at this point and have a go at those questions, please. All right, answers here. So the populations with the highest pan-IB allele frequencies would be population 1 and population 2. As you can see over here, they are pretty much almost totally coloured in for the pan-IB frequency. Okay, state the population in which the allele frequencies were closest to 0.5. I think you've got here 7, 8, 9, 10 and 11. Oh no, sorry, either 7 and 11, 8, 9 and 10 are not half and half at all, but either 7 or 11 are close to 50% each. And deduce the allele frequencies of a population in which half of the codfish had genotype pan-IA, pan-IA, so they were homozygous, and half had the genotype pan-IA, pan-IB, so they were heterozygous, and you should have got 0.75 pan-IA and 0.25 pan-IB. Pause again for me, please, and try and identify an example of 2 geographically isolated populations, and then give suggestions why the pan-IB allele is more common in population 13, which is over here, than it is in population 22, which is over there. Okay, this is a suggest question, so give yourself a couple of minutes for that. All right, so an example of 2 geography isolated populations. Basically, you could look at any of 14 to 23, okay? These ones over here, and any population between 1 and 4. So your answer could be 1 and 15 are 2 geographically isolated populations. Okay? Give suggestions why the pan-IB allele is more common. Could be anything to do with the place that they're living, okay? Cooler water temperature favours pan-IB. Interbreeding with Icelandic, oh, it is Iceland, good. And more northern populations would give different characteristics of the species. Okay, speciation, so getting to 2 different populations of species. Some old proponents of this theory thought that it was always a gradual thing.